thank you for a kind introduction, and I greatly appreciate for inviting me to this very important lecture series. And as already introduced, the title of my presentation today is the collaboration and integration of population-based and disease-based cohort studies, which is essential to the establishment of precision cancer medicine and personalized preventions. Many of the human diseases occur and are affected by exposure to various intrinsic and extrinsic environmental factors, individual genetic susceptibility to diseases, and also by their combinations. Epidemiological research using large-scale population-based cohort enabled us to elucidate what lifestyle is associated with lifestyle-related disease. The situation is also true in case of cancer, and cancer epidemiological research has provided us with various tools to conquer and control this intractable disease in an efficient and promising manner. As shown in this slide, a large-scale population-based cohort study was launched in Japan in 1990, and later a population-based genomic cohort study launched in 2011. This slide shows the trend in cancer incidence in Japan since 1975. You can see the drastic change in the incidence of organ type of cancer in the past 40 years. This change may be attributable to the change in lifestyle in Japan, also in food habit of Japanese people. We can also see the big difference in cancer incidence among various countries. Japan is ranked 27th in overall cancer incidence. This is partly because of the super-aged feature of Japanese society. This slide shows the international comparison of age-standardized mortality rate of gastric cancer. Although Japan and Korea still have high mortality rates, the rates are rapidly declining, as in the case of other Western countries. In contrast, mortality rate of liver cancer in Western countries are gradually increasing, although the rate are rapidly declining in Japan and Korea. It will be interesting to decipher or elucidate the underlying epidemiological factors of this phenomenon. Okay, now I would like to introduce and explain the history of cohort studies of cancer in Japan. As I mentioned before, the Japan Public Health Center-based Prospective study, namely JPHC, was launched in 1990 by the support of Ministry of Health and Labor and Welfare. In 2011, the JPHC Next study was launched by integrating the genomic information. Of course, in 2013, Tohoku Medical Megabank Organization started as a big project. This slide shows the study area of a number of subjects participated in the JPH study. The total number is about 140,000 subjects. This is the schedule of the JPHC study. The study was launched in 1990 for Cohort 1 and 1993 in Cohort 2. The total number of subjects is around 140,000, as I mentioned, and the response rate to questionnaires was nearly 19%. It's quite high. The baseline survey was conducted in 1990 to 1994 for Cohort 1 and 1993 to 1994 for Cohort 2. 5- and 10-year follow-up surveys were conducted to update information on lifestyle habits and health conditions. Blood samples and health checkup data were obtained at the baseline and 5-year follow-up surveys. By 2015, we had registered 28,000 incident cases of cancer, 8,000 cardiovascular diseases, and nearly 7,000 of dementia, and around 40,000 death cases. This slide shows one of the representative research outcomes of the JPHC study. long-term exposure to PM2.5, even in a relatively low levels, air pollution area is associated with significant increase of cardiovascular disease mortality by 23% in Japanese populations. Another representative research outcome of JPHC study is that the plasma concentration of polyphenol is negatively associated with colon cancer risk in Japan. Especially some of the coffee-derived polyphenols were inversely associated with the colon cancer risk as shown in this slide. I should also mention the biobank project, which launched in association with cohort studies, which played a crucial role in both population-based and disease-based cohort studies. Regarding the major population-based cohort, we have Tohoku Medical Megabank Systems, which launched after the 11 earthquake in Tohoku areas. In the case of JPHC, as I mentioned, follow-up studies among 140,000 participants, a number of subjects suffer from various diseases, including cancer. This is the slide for the past almost 30 years. Many numbers of subjects suffer from different types of diseases. Now we can, using this cohort data, we can identify some specific agent or environmental factors which is responsible to this disease onset. Using this data, we could see the population attribute factors path of cancer incidence in Japan in 2015. As you can see, infections and active smoking and alcohol drinking is a major cause of Japanese people, even for men and for women. And it is quite different from the Western countries. In Western countries, maybe alcohol drinking and the high-fat diet or food consumption is one of the major factors as extrinsic environmental factors. In the case of patient cohort, we have also two major patient cohort biobank. Biobank Japan has many, more than 200,000 patients accumulated in this data bank. And as National Center Biobank Network, NCBM, is also launched 10 years ago. NCC, National Cancer Center, is also included in this cohort. The structure and the volume of National Cancer Center Biobank is summarized in this slide. It consists of 4 sections of clinical specimens associated with detailed clinical pathological information, including more than 28,000 frozen tissue sections. Using these biobank specimens, we could identify, for example, driver oncogene alterations in lung adenocarcinoma. As shown in slides, EGFR mutation is the major driver oncogene in lung adenocarcinoma cases in Japan, which make a clear contrast in the cases of USA, in which EGFR mutation is observed only around 10%. This difference may be attributable to the difference in lifestyle or genetic background. We need to identify the specific key factors which contribute to this big difference in the incidence of driver oncogene mutations or types of mutations. Using this biobank data, we can also identify some genetic susceptibility loci affecting lung cancer risk this slide shows the data using the different biobank from the different ethnic countries. As shown in this slide, TART seems to be the common risk locus for lung cancer risk. In contrast, HADPB1, which is preferentially associated with the cases in Japanese lung adenocarcinoma with EGFR mutations. In the cases in Western countries, cholinergic receptor nicotinic alpha 3 and 5 genetic variant seems to be the major cause of cancer susceptibility genes. This also seems to be dependent upon the ethnicity dependency in the onset or in the susceptibility of lung cancers. JPHC also participate in many international collaborative studies as summarized in this table. For example, a biliary tract cancer pooling project examined exposure to smoking, anti-inflammatory drug use, health conditions such as diabetes, family history of cancer, and so forth. As you see here in this slide, Japan has the very high incidence of biliary tract cancer compared to European countries and USA. Chile also has a much higher incidence of biliary tract cancer, especially in female. This type of data is quite important to elucidate or to decipher the genetic background or environmental factors which contribute to this wide diversity in the incidence of some specific type of cancer and as from the global point of view. From this data, smoking and alcohol consumption seems to be the environmental factor affecting the incidence of high occurrence of biliary tract cancer, which is from the JPHC cohort data. We also identify the smoking and alcohol as a key factors which contribute to high incidence of biliary tract cancers. Result in JPHC are quite similar with result in pooling project. And so this means that the cohort study and also the patient-based cohort will have mutual interactions, or as for the interpretation of the data, we have a very confidence. We are going to have a very confident in terms of the understanding of the background factors which contribute to the disease onset. We also participate in the Global International Cancer Genome Consortium since 2008. This slide shows the contribution of the countries and their participation in the specific type of cancers. Japan participated in, as shown in the bottom, liver cancer, biliary tract cancer, as I mentioned, and also gastric cancer. These three cancers are very high incidence in Japan, also have very specific high incidence compared to the other countries. This is why we participate in the three major cancer projects. By participating in this global international activity, genomic activity, surprisingly, the data identified mutational signature in human cancers as summarized in this slide. As you know, the cancer developed after accumulation of many different types of genetic and epigenetic alterations, which is caused by, as I mentioned, various types of environmental or intrinsic factors, such as alcohol or alkylating agent, radiations, UV, and also some drugs and so forth. Intrinsic genetic alterations or functions of repair genes could also affect this different type of mutational signatures. This is quite useful when we consider the extrinsic factors which contribute to the disease onset. But in order to confirm whether this data is true or not, we need to conduct some experiment data using the animal models and also in vitro system, which has a mutual effect to understand or to give a right answer for this data obtained from the whole genome sequence or as international activities. Here I show the very famous data which shows the aristocratic acid as agent which is responsible for the onset of Balkan endemic nephropathy and upper urinary tract cancers. As you see here, aristocratic acid causes that, some specific type of organs when exposed to the animals, and also causes DNA DNA modification by exposure to this product, and also show the very quite similar mutation spectrum as observed in cases of this Balkan endemic nephropathies or urinary tract cancers in some specific European areas. And this combined all these data, genetic data and also experiment data, we can finally confirm that aristocratic acid is an agent which is responsible for this specific type of renal diseases. Again, the A to T base substitution caused by the aristocratic acid adduct formation, and which induces a very specific type mutation signature in the genome, which is identify the whole genome sequencing. To extend this type of research, we also analyze tumor developed in animal models by exposure to various environmental chemicals as listed in this table: BBN, PHIP, and APNH. PHIP is one of the major chemicals contained in the heated meat and fish, which humans are exposed at the daily level, daily base at a very low level. This is a representative histological feature induced in a rodent fed by feed containing food. We can see the very similar histological features with the human cases, and also mutation is commonly observed in APC and beta-catenin, which is a common target in human colon cancer cases. This data also suggests PHIP could be a candidate for human colon cancer, although some difference exists in terms of the mutation spectrum. But PHIP is a member of foodborne carcinogens, and ILC is classified this PHIP as a group 2B. It's a possible for human carcinogens. So surprisingly, when we analyze the whole genome sequence of the tumor in rats induced by PHIP, and also tumors induced by APNH, which has a very similar features and different from, it's very specific type, SBS, single base substitution type A, B, and D. Surprisingly or interestingly, and recently, some type of human colorectal cancer has a very similar mutation spectrum, SBS 94. As I show here, PHIP tumors and APNH tumors also have a very similar mutational signature as listed in this slide. PHIP is known to guanine adduct at the C8 position, as shown in the slide, and also APNH also is known to make a DNA adduct at the C8 position of guanine. And this modification is likely to induce G2T or G2A base substitution, which is compatible to this genetic analysis. Today, I described the patient-based cohort and population-based cohort. And evidence drawn by the population cohort studies can be confirmed by the patient cohort studies, and the clinical utility or biomarker candidate for early detection described by the liquid biopsy studies, for example, can be confirmed and or can be evaluated by the population-based cohort studies. This kind of mutual relationship is essential for development and implementation of personalized prevention and population-based prevention. So epidemiological and cohort studies need to be further expanded in the future to establish evidence-based and more efficient health policy at the national level. And further, from the global point of view, in order to improve health-related environments and to achieve more effective and affordable personal prevention, we also need to adopt the latest technologies such as AI technology and genome analysis, and to incorporate the wide variety of data such as health data, geographical data, and satellite data. I would like to emphasize the importance of cohort research and also the patient-based cohort. A combination of these two different types of cohort, I think we need to proceed further to identify some specific intrinsic or extrinsic or social factors which affect the onset of human diseases. Thank you very much for your kind attention.